Hello, everyone, and welcome to today's live broadcast using big data to interpret genomes for diagnostics, therapeutics, and precision medicine, presented by Dr. Rong Chen. We are excited to bring you this educational web seminar presented by LabRoots. To learn more about LabRoots, please visit us at www.labroots.com. I am Brenda Kelly Kim, and I'll be your moderator for today's event. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you'd like at any time during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your question into the box that appears on your screen. We'll answer as many questions as we have time for today. Also, please notice that you will be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located on the lower right. Finally, if you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or use the Q&A button to let us know that you're having a problem. I'd now like to introduce Dr. Rong Chen. Dr. Chen is Director of Clinical Genome Informatics at Icahn School of Medicine at Mount Sinai. His research focuses on developing databases, genome repositories, and clinical applications to interpret personal genomes for clinical diagnosis, precision medicine, predictive risk, and novel therapeutics. Prior to Mount Sinai, he led the collaborative efforts at Stanford University to drive personalized medicine and clinical diagnosis on Mendelian and complex diseases using genome and exome sequences. Dr. Chen has also helped a startup company, Personalis, which won the VA's contract for the Million Veteran Genome Project. Dr. Chen has a broad interest in translational bioinformatics and genome interpretation and has published over 80 papers in Lancet, Cell, Nature Biotechnology, Nature Methods, Science Translational Medicine. He holds numerous patents for software and databases on personalized medicine, diagnosis, and structure modeling. I will now turn the presentation over to Dr. Chen.
Thanks everyone for staying so late for the last talk with us. Yeah, thanks everyone for staying so late. Sorry for the, so today I would like to share with our recent experience in terms of interpreting hundreds of thousands of genomes to search for novel therapeutics. So we're in a hospital. We run all kinds of clinical genetic testing, including the carrier panel, non-invasive prenatal test, newborn screening test, clinical exome sequencing, and also for personalized cancer therapy test. And for healthy subject, we're also generating the pharmacogenomic report, which is not a clinical test, but also some kind of genetic testing. So we run all these kinds of hundreds of thousands of genetic tests, accumulating hundreds of thousands of genomes. And for all these individuals, we have their full electromedical record. So become a very powerful resource. So today I would like to focus on how we make use of this resource to mine all these hundreds of thousands of genomes with electromedical record to look for novel therapeutics. I'll give 3 examples for rare Mendelian disease with childhood onset, and Alzheimer disease and cardiovascular disease as examples. So to mine all these genomes, one common informatic challenge is how we filter these hundreds of thousands of genetic variants to the ones that are indeed pathogenic or drivers. And to do this filtering, we have been building this resource called reference variant store. Basically, we try to collect every single sequence of genomes, manually curating all this knowledge base, as well as all this biobank data, and building this reference variant store to filter variants to the ones that are indeed driver or pathogenic. But this is still a single variance, and we now know a lot of pathogenic variants, they are pathogenic because they cause automatic splicing, skip the exon, and so on. This is really a binding pattern. So as long as these mutations creating new patterns or break the old patterns, that will cause the exon skipping to be pathogenic. So for that one, we're building tools to predict whether a mutation will cause automatic splicing. So due to the limited time, I will focus on describing our reference variant store and how we use these tools to search genomes, look for therapeutic targets for Mendelian disease, Alzheimer, and cardiovascular disease. For reference, so to filter all these variants to the ones that are indeed pathogenic or drivers, there are several different ways. So the most common used one is some computational tools that help you to predict whether this variant is deleterious or not. So here listing for the common tools for coding variants. The DIVAS is one of my favorite because it runs for all the potential variants and runs tens of at least 20 different prediction tools and combine these results to tell you whether it's deleterious or not. And for regular one, the CADD is pretty good one. However, we all know that if you only base on these computational tools, you cannot predict these deleterious variants with very high confidence. So now people are starting to focus on one disease and so on. So the second resource, which more often used for clinical genetic testing, is to manually curate the disease variant database. So for Mendelian disease, we mostly were using this called a ClinVar. It's manually curated all these pathogenic mutations submitted by all the clinical genetic testing lab and so on. And HMG is one that are very comprehensive with the thousands of rare disease and so on. And if you want to know more detail about the disease and the detail of these pathogenic variants, you can go to Gene Review or Genetic Reference Home Reference to collect all these, to know everything about the disease and their pathogenic variants. And OpenNet is another one that tells you like all these rare disease, what their synonyms, what their clinical synopses, and it's all standard form. It can help you to compare with your other resources and so on. And for complex disease like diabetes and so on, we have been manually curating 20,000 human genetic papers, built a database called VariMat, and listing all the genetic variants that are associated with all kinds of disease like diabetes and so on. But also we collecting all the genetic variants significant associated with the traits such as lower the LDL level and so on. Besides this kind of computational tool and knowledge base, we are also interested, one very powerful tool now is because we now have hundreds of thousands of sequenced genomes, you can really take all these sequenced genomes to build a resource to know for this variant, what do I see this variant? Is the variant I see in 5% population, then that means this variant is not a pathogenic one, you can filter them out. If for the variants, I only see it in some disease cohort, that is a very interesting one, so we want to keep them. So we've been spending quite a lot of efforts to collect every single sequence genomes from healthy group, but also from all the disease cohorts to build a database called a DIVAS. So Let's compare with the existing resource. One advantage is we not only knows the frequency in healthy subject, but also knows the frequency in hundreds of different diseases. So this kind of sequencing cohort is very powerful to filter variants or understand the potential disease mechanism for the variants. However, it's still the phenotype size is still very limited, because although these individuals, they might have a severe disease, but you don't really know whether they have other disease or not. So now we come into more of this called biobank, the full patient level with full electromedical record. So for every single patient, you know their full medical histories that tell you even more information. So when you're finding a variance, you can see in my biobank, do I see patients with this variant? And for these patients, do they get cancer? Do they have other disease? Do they have very low LDL level, and so on? So if you can combine this computational tool, this manually curated disease variant database, or the sequencing cohort, as well as all the patient level full electromedical record data, you can really combine them to understand, to filter all these variants to the ones that are most likely to be pathogenic, but also to try to come up with a hypothesis that what might be the function for these genetic variants. So one work we did is we built up this resource called the DIVAS. As you can see here, listing every single sequence cohort we curated. For example, the 1,000 genomes, there are 2,500 individuals. The exon aggregation consortium, there are 33,000 genomes and so on. And this JIRA database is 80,000 individuals genomes. So we take all these in and build up a resource called DIVAS. In total, we have been curating this data from 150,000 individuals. Some of them are whole genome, whole exon, some of them are genotyping data. Not only for the healthy group, but also at least 39 disease cohorts. So to let people use this resource conveniently, we built this very simple Google-like search engines. So on the right side, you can just select all, so right at listing all the disease cohorts in our database. You can just select all of them and click into the lower buttons. Then you have all the disease cohort in your database. And then you can select either the division IDs or chromosome locations, or simply the proteins and amino acid changes, and run the queries. And it gives you this bar graph, shows you for these variants, what kind of disease cohorts we have been seeing these variants. So first, the y-axis is the population frequency of these variants, and then all the blue ones are healthy groups, and on the red one are disease cohorts. So right away, you can see for these variants, the frequency is very, very low. It's pretty much like 0.001 in healthy groups. So these variants is probably likely have some functional impact. And more interesting, you will see that although in healthy groups, the frequency of these variants is extremely low, but we do see the frequency in hypothyroidism at least increased by 8-fold, and the insulin resistance population increased by at least 4-fold. So that tells you some hypotheses that you might want to look for these variants are potentially associated with has an impact for insulin resistance and hypothyroidism. So it's a very convenient way to, by a single second, tells you what do I see these genetic variants. Is common variants or rare variants? If it's rare variants, do I see these in some very high see these variants highly enriched in some rare disease or not? So DIVAS is very useful to understand what do I see these variants. However, if you really want to search for novel discovery of therapeutics, you need to really understand the functional impact of all these variants. So in the past 10 years, we've been doing a very amazing job to identify all kinds of pathogenic mutations for hundreds of different diseases and so on. However, that we haven't found many therapeutic targets yet. The main reason is we do know these variants will cause a disease, but we do not know why these variants cause the disease. So to do that, we need to really curate a very comprehensive genetic variant database with their functional impact. So we've been turning to a literature for this resource by curating all the literature to understand what are the functional impact for each of these variants, and also curating all the protein structures. So if this variant is right in the active site, we know the functional impact of these variants, right? If it's in the substrate binding site or protein-protein interaction site, you know the functional impact of these variants. And also curating from encode phantom with these several hundreds of different tissue types, which of these variants are enhancer, active enhancers, what are their downstream substrate, and so on. And also curating all these highly conserved binding motifs like mini-meat motif and so on. So if any variants fall into these regions, you understand what are the functional impact of these variants. So for the literature, we've been downloading 30 million abstracts as well as 3 million full text and all the supplementary data. And then from that, we identify all different kinds of genetic variants. So for example, from these 30 million abstracts, we identify 600,000 substitutions, and 100,000 of them have DBC IDs. And when you turn into the full text, you get a lot more. We go to a supplementary data, we get 6 million genetic variants. And interestingly, you will see in the literature, people have been reporting at least 2,000 these frame-shift deletions, but also these 15,000 structural variants. And in total, we identified, extract from literature, we extracted 15 million genetic variants. And for all these variants, 4.7 of them you can identify their DBC IDs. And in total, we have about 4 million unique these variants. And for each of these variants, then we identify their exact alleles in total. So we identify 900,000 distinct SNP. We know the exact alleles, and also to identify these, we basically through all the signaling, but also taking this to do sequence alignment, to change the peptide codings, and then to identify what exactly this variant is. So we have all these variants in the literature. The next step is to extract the sentence describing these variants and manually annotating the function of all these genetic variants. So we're annotating the functional impact of these variants at the gene level, pathway level, cellular level, and organelle levels. So at the gene level, so for each of these variants, we manually curating whether these variants will lead to the downregulations of these genes or downregulation or upregulation of another genes, whether these variants will decrease the protein stabilities, or these variants is in the protein phosphorylation site will break these protein functions, or these variants will activating ERK pathway or deactivating a PI3K pathways, or these variants will break the binding of these proteins, or these variants that after having this mutation, you will decrease the enzyme activity by 80%, or these variants will, this mutation will cause alternative splicing and skipping the exon 10s and so on. But also whether this variant will break the protein transport and will change the substance concentrations, or this variant will be changing these drug sensitivity carriers and so on. So manually annotating the functional impact of all these variants at gene level, pathway level, cellular level, and organelle levels. Now with this functional variant database, you can then compare with all these disease variant database to understand what is the functional impact of this disease. Right. So if let's say one disease you're finding about 20 different pathogenic variants, and all these 20 pathogenic variants is all changing a protein transport, that means this protein transport is probably a key mechanism of why this mutation causing the disease. And you can then design a therapeutic, try to treat this disease. Then we want to take all these, so these are all these genetic variant annotation resource. Then we take all these together to build into some kind of infrastructures to help us to annotating all these sequencing variant data, files and so on. So we all know these. After sequencing, you have a VCF file, and every time you need to run these annotations to annotate each of these VCF files. However, you need to, every time you have a VCF file, you need to run the whole annotations. And 80 to 90% of these variants might be the same today or a month later. So you don't want to do it again and again, right? So to solving these problems, we build up these RVS called centralized variant stories. Basically, for every single variant, based on the reference genome versions, the chromosome locations, the ref alleles, alt alleles taken all together, we build a unique key for these variants. And then every single VC file, when we annotate, we automatically take all these variants, identify their unique key, which is across all studies, and build into the, compare with our RVS database to see, do I see any new variants? If I do, this new variant will be, we're creating a new variant key and accumulating there. And every night, all these variants that have not been seen before will run through all the annotations to understand what are the functional impact of all these variants, what are the population frequency of these variants in all the healthy and diseased populations. And then, so this is the annotations. And to build into some these the infrastructures, we need to take care of the sequencing for all different kind of sequencing machines, understand the quality of these variants. So we build an automated system called VAVA. Basically, all these VC files come from all different kind of sequencing machines, whether it's Illumina and PacBios. We load into the database, and we can have one-click buttons and regenerating the VC file. By that, we keep all these quality QC variables. And because it's in the relational database, now you can easily join these variants with all these annotation resource to understand what are the functional impact of all these variants. And use it to do all the post filterings and creating the clinical report, giving back to physicians, but also creating a portal to allow scientists to kind of explore all these genome data with all these genetic variants. Now, with this RVS as a tool to do all these filterings to identify these potentially driver mutations or pathogenic variants, now we can ready to mine all these hundreds of thousands of genomes with electron medical record to look for novel therapeutics. And so the approach we use is called a resilience approach. Basically, we want to learn from success instead of failures. So the modern biomedicine has been always focused on studying the failures. So for example, for humans, we always study the patients that have a disease, while we ignore the people who are just very healthy. And even for understanding the function of the genes, we'll try to knock out this gene in the systems and see what function might break, right? So these are indeed very helpful to help us understand what causing the disease, what's the impact of losing a gene. However, it's kind of counterintuitive of we should always learn from success, right? For example, if you want to live healthier longer, you should look for the people who are healthy and live longer and ask them what kind of advice they can give to you, right? So learn from these super success people is more powerful than from the people with failures. So from Starting the failure, you can learn the lesson, but starting from studying the super successful people, you can learn this powerful advice most straightforward. The reason we haven't really do much work to study these super successful people is because they are very rare, right? However, because now you have hundreds of thousands genome sequence, this kind of big data now make it possible to identify these super successful individuals. So example are first, we're looking for people who have these very severe fully penetrant mutations that you should have a very severe disease that never really should be lived to 18 years old. But although you have these mutations, there are some individuals who have these severe mutations, but they just don't show any disease phenotype. They just don't have a disease. So why? So it's one possibility is along the thousands of years, hundreds of thousands of years of evolution, human evolution, that there are some lucky individuals who just kind of live longer and have more offspring. And these individuals, they might have a second mutations that kind of buffering the effect of these disease-causing mutations, right? So if that's the case, if we can identify these resilient individuals who have buffering mutations to kind of automatically treat this disease, then we just need to figure out why they get disease. What is this buffering mutation that can turn into a treatment? And another example is we should be looking for individuals who have a very favorable phenotype. For example, individuals who have a very low LDL levels. And for these individuals, we found actually they have a gene called PCSK9 being knocked out. And then just targeting the PCSK9, you treat it, you can really lower your LDL levels. Right now, it's at least 10 billion market drug that approved last year, right? All the ones that under this immunotherapy treatment, cancer patients, and you just respond extremely well. We try to figure out why you're extremely so well, and this individual can learn from it. So I could show 3 examples of how we use this resilience approach to search for novel therapeutics. The first one is we're looking for people who have this fully penetrant disease causing rare Mendelian disease. But they are healthy, they are already adult, but still healthy. And the second example is we try to decode the individual who have a homozygous E4 for Alzheimer's disease, but they don't get Alzheimer's. And the last one is we're looking for people who have a knockout of a gene that lead to very favorable cardiovascular trait at extremely low LDL levels, and basically looking for the next, see if we can rediscover the PCSK9, and can we find the next PCSK9 for cardiovascular disease. First is for rare Mendelian disease. So the goal is we try to identify individuals who have a fully penetrant mutation lead to very severe childhood disease, but still healthy. So to do that, the key is that we need to really identify all these clinical quality disease-causing mutations. So we're lucky because we have the largest commercial screening panels in the whole countries. So we take these screening panels that and manually curating all the existing clinical tests and taking all these together to build up a very high quality these fully penetrant mutations. And then we manually read all the literatures to manually annotating for every single rare disease, which is the pathogenic mutations. And for these mutations, what are these penetrants? Like, is it 90% penetrance or just 99% penetrance? Or how severe is this phenotype? When you have this phenotype, you just cannot live to the age of 18, right? What's the age of onset? Is the prenatal born, or is the age of 2 or age of 18, and so on. So we're manually annotating the prevalence of the disease, the penetrance of the mutations, the severity of the disease, the age onset of all these disease. And then we take all these, we limit it only the disease that are childhood onset, fully penetrant mutations, extremely severe phenotype, and take these to search every single genomes being available. So far, we have searched 600,000 genomes and then try to look for individuals who have this fully penetrant mutation but are still healthy. So here shows these 600,000 individuals being searching. So as you can see, some of them are whole genome typing data, for example, 400,000 from 23andMe, 11,000 from Mount Sinai Biobank, and 11,000 from Finnish cohorts, but also this whole exome sequencing data from all the ones from BGI in China, and all 12,000 from UK, 10K, and 5,000 from Swedish, and all these, and also whole genomes from UK and also 1,000 genomes. So we take all this data and then searching these screening panels. So here shows the screening panels. For example, cardiac, we identified 190 disease, 13 genes, and 154 mutations. Then we search these and identified, very surprisingly, we identified 15,000 individuals who have this highly penetrant disease-causing mutation but still healthy. So that we manually review all this raw data to check whether these sequencing mutations is reliable or not, what's these read depth and so on. And then we quickly find there are only 303 candidates. These are past all the QCs. And then we manually examine these 303 candidates with the mutation to see how penetrant it is. And we end up only 58. Then we take these 58 candidates to do the server sequencing to confirm that this mutation is indeed there, and then manually checking their health record to check whether this individual indeed have the disease or not have the disease. So in the end, we identified 13 candidates who have these fully penetrant Mendelian disease mutations but still healthy. So here shows the 13 candidates we identified. So we identified the 3 individuals who have homozygous mutants on CFTR, but they're only 50 years old, still healthy, and do not have any report of cystic fibrosis. So these 3 are from 23andMe, and also identified 2 from UK that they have this fully penetrant mutation for sleep omics of syndromes, but they are totally healthy, and we do not observe any kind of dysphoria of the head and so on. And so, and each of these, we do all kind of quality control. So for the clinical validation side, we make sure that it passes the quality control, but also it doesn't have any classical clinical presentations or individuals whether we can. So unluckily, we were not able to recontact most of these individuals. So we really want is to bring all these individuals into our clinics and do all these lab tests, clinical tests to figure out why they didn't get the disease. Is their enzyme activity normal or lose enzyme activity and so on. But also from a genetic validation side to review all these variants, resequencing them, and so on. And then, because as you can see, initially we identified 15,000 candidates. So, but after all a series QCs, we only end up with 13. So what are these 13,000 candidates get filled out? First, so we found there are many different kind of reasons: the sequencing qualities, inaccurate database information, or this mutation clinical review, or this individual clinical reviews. So these list all the reasons and how many individuals they get filled out. So first you will see there are about 7,000 of them they filled out because of the sequencing quality. there are still a lot of the sequencing errors, especially this low abundance or the variants that are doesn't fit into Hardy-Weinberg and so on. But also there are some variants. We actually identified 15 variants. That their frequency, although clinically believe they are fully penetrant disease mutation causing Mendelian disease, however, when you search against 600,000 individuals, you found that their frequency is very high. Actually, 0.5% in population has these mutations, which means these mutations is not really disease causing, it's a polymorphism, which leads to about 7,000 individuals get removed. But also like pseudo deficiencies and so on. And from mutation clinical review, we also identify, so for example, there are 6 mutations that we know that if you are heterozygous, you're causing very severe disease. But if you're homozygous, actually, you don't have any symptoms. So these ones should also be removed. And when you manually review these single candidates, especially when we do the sentinel sequencing confirm, we found there's still quite many individuals, actually including 15 individuals that when you do the sentinel sequence, sentinel sequence confirm, you found actually they are not really homozygous, they are heterozygous, so they get removed out. But also we identified 10 individuals that their sequence is confirmed, but when you check in their health record, they do have some phenotype. Although they didn't get diagnosed for the disease, but they do have some phenotype that is more of a mild form of the disease. So for this individual, we cannot say they are resilient individual. We also remove all of them. So that in the end, we get is 13 resilient individuals. And this graph just shows, it's a nice graph, shows what our panel is, and also how many individuals we searched for every single variance, as well as whether the standard whole exome sequencing, whether it covers well for all these pathogenic mutations. And in the end, we identified these 13 candidates. So, as I said, so we already identified 13 resilient individuals. They should be a very powerful resource to identify why they didn't get a disease, is there any potential buffering mutations that protect them getting the disease. However, we haven't been able to bring these 13 individuals back into the clinic because of the lack of concerns and so on. So, but we do one product coming out of these studies. We have a high quality mutation panel. Now you can take this to search in your genome to see whether you see any resilient individual or not. So in this website, we write the software, and it's totally public resource. You can take this software and run in your genomes to see whether you can identify any resilient individual or not. Then it was published in Nature Biotechnologies, and but like I said, we do want to be able to not only identify resilient individuals, but also bring them back into clinics to do all the clinical testing to figure out why they didn't get a disease. So to do that, we launch a prospective studies to do resilience project. So we take all these resilient genes and mutations, but also all the rare Mendelian disease genes, as well as the genes for cancer and cardiovascular and pharmacogenomics, put all these together. Not only these disease-causing genes, but also the ones that outside the gene regions, which we know they have identified from all these human genetic studies have an impact. We take all these regulatory variants together and build a very comprehensive super panels. And so now we're running these super panels, launch these all plan to sequence 100,000 individuals to look for resilient individuals. So launch this prospective resilience project is totally free. In this website, resilienceproject.me, you can sign up, and it will send you an e-consent, and you can consent, and then you will receive a swap kit. Follow the instruction, you swap your kit and ship it back we do all the sequencing and run the analysis, identify whether you are a resilient individual or not. So now I want to switch from Mendelian disease to Alzheimer's. Like I said, this resilient project is very powerful, but we do want to decode these resilient individuals to see why you didn't get a disease. So we turn to Alzheimer's disease, and the reason is for Alzheimer's, there's an APOE E4 homozygous. Your risk, if you're homozygous E4, then your risk for Alzheimer's will increase by 12 times. However, you can still identify very large number of individuals who are resilient, so give us enough power to decode them. So to do this, we collect 6 large Alzheimer sequencing data cohorts. First, from Mount Sinai, we're looking at 13,000 samples. From them, we identify 93 individuals who are APOE E4 homozygous, including white, African American, Hispanic. And we also identify 5 different resources. The Geisinger Health Systems has 7,000 individuals and 132 homozygous E4 individuals. This GBAT identified 103 homozygous E4, ANI identified 91 homozygous E4, ADGC identified 456 homozygous E4 individuals, and ADSP has 145 homozygous E4. So taking all together, we identified 1,000 APOE E4 homozygous individuals, and 570 of them already been diagnosed with Alzheimer's, and 190 of them are healthy, and their age is always above 60s. So first, we take the Mount Sinai cohort to do a discovery. So we compare. So we're looking only at the individuals who are APOE E4 carriers, and compare the ones who get Alzheimer's versus the ones who don't get Alzheimer's. And we identified a loss function mutations, basically a knockout of the gene that have a protective effect on Alzheimer's. And it has been shown in both, has a protective effect in European, African, Hispanic. But the p-value is not very high, odds ratio about 2. So we take these values and checking 4 different Alzheimer's sequencing cohorts. So this is more of a meta-analysis. Basically, each line is one Alzheimer's sequencing cohort, and every single individual is a homozygous E4. And if you see the dot on the left side, means this variant has a protective effect in all the APOE E4 homozygous individuals. If you see the dot on the right side, means this variant will increase risk for Alzheimer's. As you can see, the variant we identified from Mount Sinai cohorts, it has been replicated in all 4 European cohorts, the GBAT, Geisinger Health System, ANI, and ADGC. And the odds ratio is very similar. Basically, in all the individuals who are homozygous E4, if you have a knockout of this gene called Cas7, you will have a protective effect about 2. And it's a similar effect size across all studies. Interesting, if you're looking at the individual who are non-carriers of E4, you can see we don't see any protective effect for this variant. So this paper will be published in BMC Genomics in a few weeks. So for this, the variant we identified is a loss function variant in the first exon, and but we need to validate whether this variant indeed leads to the loss function of Cas7. So when we pull out the tissue for prefrontal cortex and so on, and then check it for EQT, we do see that if you have this variant, the expression of Cas7 will be significantly decreased. So that means this variant is not only a phase shift loss function variant, but from MI levels, we do see it leads to the lower expression of Cas7. Then, as I showed, that we build this DIVAS resource to help you to identify the frequency of any genetic variants across all the healthy and disease cohorts. So same thing, we take these variants and search in the DIVAS to see what we find. So as I said, the y-axis is the frequency of these variants in all different cohorts, and the blue one are healthy cohorts, red one are the disease cohorts. First, you will see this variance is a common variance. It's about 19 to 15% healthy individual all carry this variant. And interesting, you will see is in the familial ALS populations, the frequency is extremely low. So that means probably this variant also has some protective effect for familial ALS. That's why you didn't really see many patients has this variant, because if you have this variant, you have a less chance getting familial ALS. Then we're looking at all the healthy groups. You notice is the frequency in African American is about 42%. It's much higher than the 32% in European. So one thing we do know about APOE4 is if you're European and you have an APOE4, your risk for Alzheimer has been significantly increased. However, if you are African, although you have an APOE4, your risk for Alzheimer is only increased a little bit. So maybe because Africa has more of this, particularly of this non-primary mutation of caspase-7, which leads to the African American who carry APOE4, but still have a less likelihood of getting Alzheimer's. So what are the functions, what's the mechanism of this caspase-7 to protect Alzheimer's? So we do know in all the Alzheimer's patient brain, this caspase-7, it is indeed activated. And when you knock out this caspase-7, you will attenuate microglia activation, which might be the mechanism for the caspase-7 to be protective for Alzheimer's disease. And so last, I want to describe this for looking for therapeutics for cardiovascular disease. So this is our Mount Sinai biobank. We have 2 million patients with a full electronic medical record, including 16 million patient visits, 36 million ICD-9 code of diagnostics, and about 94 million of medications, and 182 million of these blood test results. So our goal is, as we know, each of us all have some gene get knocked out. The goal is to identify for if you knock out a gene, do you have an extremely favorable lower LDL level or not, right? So we have 11,000 patients that we have the genome sequence as well as their full electronic medical record, including all the blood test results, LDL level, and so on. So from them, we identified 3,000 genes that have to knock out individuals in our biobank. So for each of these genes, we identify an individual who have a knockout of the gene put into the trace group, the individual who don't have a knockout to the control group, and then we compare when you knock out these genes, do you have a lower LDL level or not? And we do know this individual taking statins and many other medications that may impact LDL level and so on. So for every single trait, we identify all the medications that might have an impact and remove all these individuals, the measurement that is after treatment. So these are the 11 traits we've been focused on. So for LDL level, HDL level, total cholesterol, glucose, triglyceride level, and so on. And so here's our results. Basically, each line is one trait, each one gene that get knocked out, and you can change this trait level. So for example, the first one shows the when gene get knocked out, we identified 23 individuals who were knocked out of this gene. And among these knockouts, you will lower your LDL level from 102 to 62, basically cut half. And this one is a PCSK9, a 10 billion market drug. But also we identify many other genes that get knocked out and change your HDL level, LDL level, and so on. So here shows the result for PCSK9. The y-axis is the LDL levels. On the left side is the individuals who get knocked out of PCSK9. On the right side is the individual who have half PCSK9. So you can see for all the individuals, when you get knocked out of PCSK9, your LDL level is extremely low. And here is another example for APOC3 that we have a knockout APOC3, you can see your triglyceride level was significantly lowered. But this is only shows that the extremely that we knock out APOC3, right? So we turn to a second data set. We have 500 atherosclerosis patients. Each individual, we know their triglyceride level, LDL level, and so on. And each, we have 9 different tissue types. And every single tissue type, we run the whole RNA-seq. So this graph shows each dot is one individual. And in the liver tissues, when you have low expression of APOC3, your triglyceride level is also significantly lower. So now we not only know that you knock out APOC3 can lower triglyceride level, but also knows when expression level APOC3 decrease, you will see triglyceride level significantly decrease. And so here is a new gene we identified. We have a knockout of these genes. You can lower your triglyceride level from 145 to 108, and fasting glucose can lower from 111 to 98. And then we do identify a drug for these genes. So we take all the mice and giving these treatments, these drugs for 3-day treatments. So each dot here is one mice. On the left side is all the controls, and on these two shows 2 different genes. So we knock to give a treatment for 3 days. You can see all the mice after giving the treatments, you can significantly lower the fasting glucose level. So in summary, that we have been manually curating all 100,000 individual genomes, identified these 400 million variants with their what we see in the healthy group or disease populations, but also manually curating from 30 million publications for 4 million distinct variants of their functional impact. And we use these to search against the genomes with electronic medical record. We'll be able to identify certain individuals, resilient individuals who have free penetrant mutation but are healthy. But also we found a knockout of caspase-7 can protect Alzheimer's disease in APOE E4 carriers. And we're finding a novel gene when you knock out, you can lower the glucose levels. So we take all these into a single panel to run large-scale, low-cost sequencing. And this is my team who did all the work. And this is my contact, and that ends my talk. Thank you for staying late with me, and if you have any questions, please free to ask.
Thank you very much, Dr. Chen, for bringing that important information to us. Before we get to our questions, I just want to remind our audience that questions can be submitted by clicking on the green Q&A button on the lower left, submitting your question on the box that appears on your screen. We'll get right to our first question. An attendee asks, what is the frequency of de novo versus inherited variants?
Yeah, that's a very good question. I don't have an answer right now. It was pretty low. I think there is some publication showing, yeah, I don't remember the exact number, but I think you're looking at probably 20 de novo mutations per person. So don't call me on that. There's a paper you can about this.
Okay. Thank you. Our next question. Is there any example of applying DIVAS to individual clinical patents, genetic variants data being used in a real clinical setting?
So we do have including a DIVAS into a system called ClinLab Genetics. That is a whole software has been used in the clinical setting at Mount Sinai to analyzing, to manage the whole process of a clinical testing for clinical whole exome sequencing. So that paper has been published last year, and it's free download. You can download the software to run in your clinical settings. And at Mount Sinai, every single clinical genetic has this software installed and help us to manage this whole process the review of these variants, but also managing the whole process as which variants should be sent to the central sequencing control, how to create a report, and so on. And one feature of this software is in a real clinical settings, you have different people, whether it's a director or administrator or genetic reviewer, you will have seen different interface and so on. But for more detail, you can search for Clean Lab Genetics software or just visit my web website.
Okay, our next question. Have you looked at the correlation of all the gene variants of specific disease along with gene networking of similar clinical phenotype?
Can you repeat the question? Oh, sorry.
Do you need me to repeat the question? Have you looked at the correlation of all the gene variants of specific disease along with gene networking of similar clinical phenotype?
So short answer is no, I have not. One thing I'm planning to do is we have all the patients cohort here, and we know they have this disease causing variant. We're specifically trying to look for the individual who have these variants, but because they lacking some phenotype, they get misdiagnosed, they lack of a clinical diagnosis, but they might have this phenotype very close to the rare Mendelian disease. And then when you're looking at this gene network, I think probably not only need to bring in these genetic variants, you also need the RNA level, this kind of biomarker as well. So we have been using the RNA data building the network and try to plot all these genetic variants onto this network. That kind of help you to see which are potential drivers, potential therapeutic target.
Okay, I think we do have another question. Somebody would like the web address or other way of accessing the software you mentioned?
Go to my, if you go to the website of my lab, rongchenlab.org, you will see the download of the software. You can download and search for resilience, or the Clean Lab Genetics you can use to run for clinical whole exome sequencing. And for my website,
Okay, I think that's all the questions that we have right now. I'd like to thank Dr. Rong Chen for bringing this information to us and taking time to be here with us today. And I want to let our audience know that this webcast is going to be available for on-demand replay. You'll be receiving a notice from LabRoots as to when it's available for replay, and it will stay up through November of 2016. We would invite you to forward that email to any colleagues who may have missed today's live presentation. Dr. Chen, do you have any final thoughts for us?
Yeah, thank you. Thank you for staying with me for the last talk, staying so late. And I would like to thank Brenda and LabRoots for making this available. If you have any more questions, feel free to send me an email.
Thank you very much, Dr. Chen, for your time today, and we thank you for logging in and listening to this presentation. We hope we see you again. Goodbye.